So, small molecules are transported into and out of the mitochondria via transport proteins or transporters. This is how stuff gets in and out. They have two main conformations: open out and open in. Let's draw those so we can kind of understand what's happening here. So, open out in my cartoon drawing will look something like that. And open in will look like the opposite. We can imagine that here's the matrix, and this is the inner membrane space. And so these proteins have kind of this channel going through them, and either end of the protein could be open. So this protein, kind of like this, can open either out or in. And molecules can kind of make their way through the center of these channels. Transport of these small molecules is mostly mediated by local concentrations. So these molecules are falling down their concentration gradients from areas of higher concentration to areas of lower concentration because this is thermodynamically favorable. Let's talk a bit more about these transport proteins and their structure. So these proteins generally have two binding sites for their ligand. Ligand is a word that is similar to the word substrate, although generally we do not perform chemistry on ligands, right? Ligands are more the things that bind to binding proteins. So that's another word for us to use. What does this look like? So I'll draw just one version of these proteins for now. And so the two binding sites will be kind of symmetrical. We'll have one that'll be the in site and one that'll be the out site. So you can imagine some type of small molecule that's being transported will go and bind the first binding site. That'll cause some type of conformational change where the shape inverts. And then that molecule can kind of make its way to the second binding site and out the other end. And so the transport of this molecule is facilitated by kind of binding first to one site and then binding to the other site, along with the conformational changes that happen. There are three different types of transport that these proteins do: uniport, symport, and antiport. Uniport is the transport of one molecule. Symport is the transport of two molecules in the same direction. And antiport is often the transport of two molecules in different directions, or I should say opposite directions. And we'll see a few examples of this. The first transporter we're going to talk about moves pyruvate into the mitochondria so that that pyruvate can enter the citric acid cycle. It is moved in by a protein called pyruvate translocase. This is a symport with a proton coming in across this concentration gradient. Let's draw this out. So here's our membrane. Let's make sure we label this time because it's going to be important for us to know which way pyruvate goes. And we should understand that there'll be a conformational change where this kind of out conformation changes to in conformation, but we don't need to draw that. So what's happening here is we have pyruvate going through the channel along with a proton. So this symport of pyruvate and a proton into the mitochondrial matrix facilitates the citric acid cycle because we're getting the product of glycolysis, which is pyruvate, into the matrix to enter the TCA. It is powered by a proton falling down its concentration gradient. As we will learn next time we're together, even though I know most of you already know this information, there is a huge concentration gradient in the inner membrane space that we call the proton gradient, right? There are so many protons here that one falling through this transporter powers the translocation of pyruvate. That is the pyruvate translocase. So in addition to the citric acid cycle happening in the mitochondria, we also have to think about electron transport, and we'll talk much more in detail next time. These electron transport proteins are embedded in the inner mitochondrial membrane. And they cause this proton gradient, where the concentration of protons is extremely high in the inner membrane space. Now, after the electron transport chain, as we'll also learn coming up soon, that proton gradient is used to synthesize ATP from the substrates ADP and 1-phosphate. For all of this to happen, ATP that is synthesized in the mitochondria probably needs to leave the mitochondria to go do whatever it needs to do to power the cell. And then ADP and phosphate need to come into the mitochondria to be converted to ATP. Well, welcome the ADP-ATP translocator. This protein I have some nice structures of, so let's talk about those structures. It is a dimer. We have some, oops, I didn't actually mean to draw just then. There we go. So we have shown here kind of like the secondary structures made more visible, right? We see alpha helices and they're kind of like intertwining with each other. On the right hand side, this is a more space filling model where we can see the overall shape. I also have for us a top down view. And so you can see in these transport proteins that this channel exists where a small molecule will kind of enter and make its way through the transporter. This transporter does antiport, so one thing comes in, another thing leaves in opposite directions. It's pushing ATP out and pulling ATP in. We'll talk about this again on the next slide when we draw it, but this transporter is powered by the proton gradient in kind of a roundabout way, in that it's doing electrogenic antiport. If it's sending a -4 charge molecule out and a -3 charge molecule in, then that is a net charge difference of 1. So it's sending 1 negative charge out. We'll talk about that again as I draw. Okay, folks. Let's draw our protein again. We got the matrix. We got the inner membrane space. Okay, so remember, we need to get ATP out, ADP in. So let's just draw that. ADP going in. ATP, when it is synthesized, needs to be sent out to be used by whatever pathways need to use it. that leaves us with a charge outside of -4 and a charge inside of -3. That cancels out to just a net of -1 on the outside. Remember, folks, this proton gradient exists. And that proton gradient is an electrochemical gradient. So in addition to that proton being there, we also have to look at the charge. So the transport out of a net -1 charge is balancing out this massive positive charge that's building up in the inner membrane space. So in a way, the proton gradient is powering this transport because it's dependent on that charge, the proton gradient, and kind of balancing it out by sending out that -1. Now, I told you that when we're synthesizing ATP, we need to use a DP and a phosphate to make ATP. So how does phosphate get in? And this will be the last example today to wrap up the video. We use the phosphate carrier. It is, again, a symport, and it sends both a molecule of phosphate and a proton across the membrane. So it is driven by the proton gradient. And so we should be very comfortable with this by now. Here's the inner membrane space, the matrix, and we need to get this phosphate into the matrix so that we can do synthesis of ATP. That phosphate is pulled through with the help of a proton, falling down its concentration gradient from an area of high protons to low protons. So this should show us, folks, all of these three examples of transporters, that the proton gradient is incredibly important. I think probably all of you already know it's essential for synthesis of ATP in our bodies, but I want to recognize that it's used for a variety of other functions, and we only looked at 3 today. That wraps up our time today. We talked about how different metabolic pathways occur in different locations within a cell, separated out oftentimes by organelles. We talked about membrane transport and how small molecules get in and out of the mitochondrion. We went through a few specific examples. First, we talked about how NADH sends its electrons into the electron transport chain, but doesn't actually enter the mitochondrion. And then we talked about 3 different membrane transporters that move pyruvate, ADP, and ATP, as well as phosphate. That's all, folks. I will see you in class.